Okay. Good morning, everybody. The first thing that I wanted to talk to you guys about today is the fact that I did make a couple of announcements this week, so if you haven't had a chance to take a look at those announcements yet, I would go ahead and do that. The big thing that I wanted everybody to know is that I did grade some of your exams already, but at this point I still have a couple of more steps to take in terms of circling back around after everybody's individual grades are uploaded and determining which of the questions from the exam the majority of people didn't get credit on, and then I will give everybody points back for that. At that point, if the exam is still not where I want it to be, I'm going to go ahead and apply a scale, but right now everything looks to be in pretty good shape. So I'll be in touch with you guys with updates on, how far along we are in that process. All right, so last time we met, we started the lymphatic and immune system, and so today we're going to go ahead and finish up that chapter. I think what I'm going to do is probably, depending on how quickly we move through, I'll talk about the adaptive immunity, then I'll do another presentation on cellular and humoral, and then one final one on immune system disorders, depending on how long it takes to get through these slides. All right, so last time we met, we talked about innate immunity. So hopefully you have already watched those lectures. Otherwise, this one's going to be a little bit more difficult to follow along with. But today we're going to be talking about adaptive immunity, which is your third line of defense, okay? So innate immunity makes up the first and second lines of defense. Adaptive immunity is what makes up your third line of defense. And it's characterized by three aspects. So we talked about these aspects when we talked about innate immunity, and basically they are that adaptive immunity has effects that are systemic. So what that means is your adaptive immune response is going to be observable throughout the entire body, as opposed to the innate response, which would be comparable to something like a bug bite or maybe a splinter, where you get a small local reaction, but you don't really have any systemic effects. But we did talk about last week the one exception to that rule is the fever is actually part of the innate response, and that is a systemic response. Other than that, adaptive immunity is what is responsible for systemic responses. In addition to that, the effects of adaptive immunity are also specific. So what that means is that we don't get this general response that sort of hits a widespread variety of pathogens. Instead, its response is directed at a specific antigen. Okay, so while a fever doesn't really have much specificity as part of the innate response, in adaptive immunity we have these lymphocytes here that will clone themselves to basically be directed at a specific antigen. So we'll talk about that as we move through today. And then finally, with adaptive immunity, the last aspect is that it's involved in memory. So what that means is when we see an antigen or a pathogen for a second time, with the adaptive immune response, we basically mount a secondary response so quickly that there's often no noticeable signs of illness. Okay, so we don't even realize that we've come into contact with this antigen again because our body takes care of it so quickly. So there's different forms of immunity, cellular versus humoral, and we're going to brush on them quickly here, and then we're going to go into each of them in more depth in the next couple of chapters. There's also active versus passive immunity, and then those can be described as natural versus artificial. Okay, so looking at them individually, cellular immunity is different from humoral immunity because cellular immunity employs T lymphocytes and humoral immunity employs B lymphocytes. So T lymphocytes in cellular immunity are really important because they're the only lymphocytes that are going to directly attack foreign cells or infected host cells, while humoral immunity, on the other hand, does not directly destroy pathogens, but instead uses antibodies, which will either neutralize the pathogen as opposed to killing it, or it will just mark it for destruction by other cells. So cellular immunity, using T lymphocytes that directly attack foreign cells or infected host cells, are going to target pathogens where they're not accessible to antibodies. So that's going to be places like inside the cell. Intracellular viruses, bacteria, yeast, and protozoans. Cellular immunity also targets parasitic worms, cancer cells, and cells of transplanted tissues or organs. Humoral immunity, on the other hand, that uses B lymphocytes, which produce antibodies that don't destroy pathogens but instead mark them for destruction or neutralize them. This type of immunity is going to target mainly extracellular stages of infectious microorganisms. So that's going to be viruses, bacteria, yeast, and protozoans that are outside of the cell. It also targets toxins, venoms, allergens, and transfused red blood cells. So basically, in a nutshell, cellular immunity is going to directly attack foreign cells in places that they can't be reached by antibodies. Well, humoral immunity is going to use antibodies to flag those pathogens or microorganisms in places where they are accessible to antibodies, so outside of the cells. Active versus passive immunity. So active immunity is different from passive in that it involves memory to provide future protection. So this is what happens when your body makes its own antibodies and T cells against a pathogen. Here we have the example of a vaccine. So you get this vaccine, and we'll talk about vaccines in the next couple of slides. But you get this vaccine that's basically full of all of these dead or weakened antigens, and your body responds to that by producing its own antibodies and T cells, which provides you with memory so that in the future, when you do come into contact with this antigen again, maybe the living form, your body already has produced all of these antibodies and it has this memory of this antigen to help mount an immune response really quickly. On the other hand, passive immunity doesn't involve memory to provide future protection. And what happens in passive immunity is that the body basically gets these antibodies from another person or another animal that's already immune to the pathogen. And so this type of immunity lasts for a period of weeks. An example of this would be basically when a newborn calf is born, or any newborn animal, and it drinks colostrum or breast milk from its mother. It's going to acquire some antibodies from the mother that it wouldn't have otherwise. This is especially important for baby calves because of the fact that when they're in utero, the placental material between the mother and the uterus is so thick that they're not able to pass antibodies back and forth to the fetus, and so drinking this colostrum is really essential for survival. If they don't get these antibodies in the form of passive immunity, then they're really likely to die or at least become really, really sick. Okay, natural versus artificial immunity. So both of these, both natural and artificial immunity, can either be active or passive. Okay, so we just talked about active versus passive. Active is what's involved in memory and future protection. Passive doesn't involve memory or future protection. So let's talk about natural active immunity first. So this would be basically if you encounter some kind of antigen through natural exposure. Let's use coronavirus for an example, and you start to produce your own antibodies. Okay, so you come into contact with somebody who has coronavirus, you contract the virus, you get sick for 2 to 4 weeks or whatever it is, and then after that, if you come into contact with the coronavirus again, your body has already mounted all of these antibodies to basically combat the virus. So that's natural active immunity. In comparison to artificial active immunity, this would be like the example we talked about if you got a vaccination. So that's the artificial aspect of it. You're basically it's active immunity in that you're producing your own antibodies or T cells, but it's artificial in that it came from a vaccination. Okay, so again, you get this vaccination full of dead or weakened antigens, and your body mounts all of these antibodies to prepare you for some sort of secondary encounter. So moving back to natural immunity, in the passive form, This would be like the example that we talked about with the baby calf acquiring antibodies through the colostrum of the dam or the mother cow. So this is a temporary immunity that you get from basically providing yourself or providing an animal with antibodies that came from another person or another animal. So a fetus can acquire antibodies through the mother's placenta in utero, or again after birth through breastfeeding. And then we contrast this to artificial passive immunity, which is basically immunity that you get from injecting yourself with serum that comes from a person that already has antibodies to a certain pathogen. So we use this type of immunity in emergency treatment of things like snake bites, rabies, or tetanus. Okay, so a little bit more about vaccination. I know we talked about this already, but vaccination is what produces artificial active immunity. Okay, so basically what happens is a vaccine carries these really weakened or dead pathogens. And what happens is when those pathogens are introduced to your immune system, they'll stimulate an immune response, but you really have no discomfort or very little discomfort to the disease itself. And what we can do is give booster shots periodically to re-stimulate that immune memory. So here we have the vaccination, and then you're producing antibodies to this weakened or dead antigen. And then down here, after vaccination, if you're met with that pathogen in real life and it's alive and living and well, now your body has already produced all of these antibodies to target that antigen. So vaccination has been a really great thing in reducing many of the diseases that were hugely prevalent in children worldwide, but recently there's sort of been this movement of more holistic approach to medicine in terms of anti-vaccination superstitions and things like that, and so people have been shying away from vaccines more than they had been previously, and so a lot of these diseases that were eradicated completely, we're starting to see them resurface again, and so many people are continuing to die from diseases that could be prevented by vaccination. So antigens we talked about already. This should be a little bit of a review for you guys. We started talking about them back when we talked about blood transfusion and red blood cells in the last section of the lecture. So antigens, again, are just any molecule that trigger an immune response. So they're what basically mark your cells as distinguishable from foreign cells. And they can be in the form of free molecules, so like a venom, like a bee venom or a snake venom, different toxins. Those can be basically free-floating antigens, but they can also be components of plasma membranes. And so let's say this cell here is a bacteria that is invading our body, and we have all of these different little shapes up here. Those represent antigens. And so that's how your body distinguishes all of these different little shapes, is how your body distinguishes, is this my cell or is this some other foreign cell? And so those antigens are going to be composed of complex molecules. So they're never going to be something super simple like glucose. They're always going to be something much longer like proteins, polysaccharides, glycoproteins, glycolipids. And the reason for that is because they have to be unique. Okay, their uniqueness is what allows the body to distinguish, are these my cells or are they cells of something foreign? Epitopes and haptens, so there's a lot of new language that you guys will have to learn for this section, and so what I did, I think probably somewhere around the end of the third slide, I combed through and I picked out really important vocabulary words for you guys to focus on. Epitopes is one of the really important vocabulary terms for you to know, and it's basically just the active region of the antigen. Okay, so here we have an antigen and it's binding to an antibody. So here's our antibody, here's our antigen. Epitopes are this region of the antigen that make the antigen active. So they're going to be the binding site to the antibodies. So they're the part of the antigen that stimulates an immune response. And one antigen can actually have several epitopes, so we can stimulate the production of many different antibodies just even with one pathogen based on the number of epitopes that that pathogen has. Haptens, I don't have any pictures here, but you can use your imagination based on my explanation. They're basically just incomplete antigens. Okay, so they themselves cannot stimulate an immune response, but what happens is they bind to some sort of macromolecule on the surface of a host cell. So let's just say they bind to like a glycoprotein or something. And once they bind, then they basically become long enough to be complete. And so they make a complete antigen once they bind to a macromolecule that's on the surface of the host cell, and that's going to stimulate an immune response. So penicillin is just one example of a hapten that behaves that way. Antibodies should also be a review for you guys. They're otherwise known as immunoglobulins, which is why you see Ig down here. Antibodies are proteins that you can either find in membranes of your B lymphocytes, or you can find them floating freely, just sort of dissolved in your body fluids like plasma or breast milk. And there's 5 different classes: IgA, IgD, IgE, G, and M. So in most species, IgA is the really important one in serum and also in mammary secretions. So colostrum, for example, in humans and most other species, IgA is going to be the really dominant antibody, but for cattle, IgG is the most important. All right, so the basic structure of an antibody. An antibody is something called a monomer, which basically just means we can hook a whole bunch of these together. We can put 2 of them together to make a dimer. We can put a whole bunch of them together to make a polymer. And so they're basically made up of 2 heavy chains, which we see on the inside here, and also 2 light chains. So one here and one here. And the difference between those 2 chains, as we can visibly see, is that the heavy chain has about twice the amount of amino acids as the light chain. So there's a disulfide bond here, which gives the antibody this Y or T shape. And then in yellow here, we see what are called variable regions. So these are the regions of the antibody that give each antibody its uniqueness. All right, and there's 1 on each of the 4 chains. So 1 on this heavy chain, 1 on this heavy chain, 1 on this light chain, and 1 on this light chain. And so what happens is this variable region from this light chain, and this variable region from this heavy chain come together to form a binding site for the epitope or the active part of an antigen. Okay, so if we go back here, we see the epitope binds here. This is the variable region of this heavy chain, and this is the variable region of this light chain. Together, they form the binding site for the epitope of the antigen. All right, I know it's a lot of vocabulary, so. All right, lymphocytes and adaptive immunity. So last lecture we talked about lymphocytes, specifically in innate immunity. We talked a lot about natural killer cells, which are these guys right here. And we just kind of brushed on T lymphocytes and B lymphocytes because we were talking about innate immunity, and these guys are both involved in adaptive immunity. So today we're going to focus a little bit more on T and B lymphocytes. So starting with T lymphocytes, basically, just like all other formed elements, they are produced in the red bone marrow and developed from hematopoietic stem cells, which everybody pretty much got that correctly on the exam so far. And so what happens is after these newborn cells come from the bone marrow, they're going to enter into the bloodstream and travel to the thymus, which, if you remember, is the endocrine and lymphatic gland that exists right above the heart. And so within the thymus, they're going to mature into immunocompetent T cells, which is what you see here. So immunocompetence basically just means they have the ability to respond to a pathogen and produce an immune response. All right, so what happens inside the thymus that makes them immunocompetent? The first process is called positive selection. Okay, so basically what happens is these immature T cells arrive to the thymus, and within something called the thymic cortex, they'll go through this process of positive selection. So what positive selection does is it's basically a way to ensure that these T cells have the receptors that they need to recognize foreign pathogens, foreign antigens, or antigen-presenting cells. So what you see in this picture down here is a T cell is being presented with an antigen, and if it binds, then it will get this basically survival signal from epithelial cells. Okay, so it receives a protective signal from the epithelial cells within the thymus, and it survives. All right, so again, the T cell is presented with an antigen, and if it binds, then it survives. So that's positive selection. Cells that don't recognize antigens like this, or don't recognize antigen-presenting cells, they have a chance basically to retake the test. They can reshuffle their DNA and try again. And at that point, if they're still not able to recognize the antigens or the antigen-presenting cells, then they'll die. And they'll die by apoptosis, and then they'll be consumed by macrophages. So the next process that takes place to make sure that our T cells are immunocompetent is called negative selection. And so we get this smaller pool of T cells that have passed positive selection. They're going to go to the medulla of the thymus and undergo negative selection. So what negative selection does is it's basically a way for our body to make sure that these T cells don't react or react too strongly to antigens of our own bodies. So we get a T cell presented with a self-antigen, and if that T cell reacts too strongly or it binds, then it's going to be killed by apoptosis. All right, so again, if a T cell is presented with an antigen from our own body and it binds and reacts to that, then it's going to be killed. Okay, so after this, after we go through positive selection here, making sure our T cells can bind to foreign antigens, and negative selection, making sure our T cells don't bind to our own antigens, we only have about 2% of T cells left surviving. And what happens is those T cells are going to be cloned, so they'll reproduce into more T cells, and these guys are considered naive, part of the naive lymphocyte pool. So they're basically like soldiers that have never been to war. They're immunocompetent lymphocytes, so they have the capacity to respond to an antigen, but they've never encountered any foreign antigens. So what happens is they'll leave the thymus after they're produced. They'll go into the lymphatic vessels and the blood vessels, and then they'll travel to different lymphatic tissues and organs, and that's where they'll stay long term. So B lymphocytes go through pretty much the same process. They're not as well understood as T lymphocytes, but they do go through pretty much the same process as far as we know. And so I sort of just summarized all of it on this one slide, but basically what you're seeing here is those B lymphocytes, instead of going to the thymus to mature, they're going to just mature within the red bone marrow. And a normal adult produces about 50 billion cells per day of B lymphocytes, but only 10% of those are going to survive to be entered into circulation after positive and negative selection. Remember, we talked about here only 2% of the T cells survive positive and negative selection in the thymus. The B cells, about 10% of the 50 billion that we produce, will survive after positive and negative selection. Then those surviving B cells will be cloned and again dispersed throughout the body to colonize the exact same tissues that those T cells colonize. Antigen-presenting cells, we talked about these guys during our last lecture, but just a little bit more information on how exactly they function. So antigen-presenting cells are just cells that basically digest antigens, break them up into little tiny pieces, and then expose the important parts of those antigens on their cell surface so that other cells can recognize them. Okay, so here we have a macrophage, which is one of the antigen-presenting cells. So B cells, macrophages, dendritic, and reticular cells are all cells that act as antigen-presenting cells. So here we have a macrophage that you can see in this example. and basically it's going to consume an antigen, chop it up into little pieces, and then expose those important pieces on its cell surface so that T cells, like this one here, can recognize it and stimulate an immune response or an attack. So the way that it's able to do that, or to present these epitopes, which if we remember are the active part of the antigen on their cell surface, is through something called MHC proteins. So MHC proteins are what we see right here. They're proteins that exist on the surface of an antigen-presenting cell like a macrophage. They're shaped like little hot dog buns, and they're structurally unique to every single person except for identical twins. So they are also, much like antigens, what are responsible for labeling every cell of your body as belonging to you. And they're involved in the antigen processing, because basically what happens is, here's an antigen presenting cell, this whole big thing. It's going to consume an antigen. So once the antigen has been consumed, it's going to be chopped up into little pieces. Then the important part of these little pieces, the epitope, the active part of the antigen, will be exposed on the MHC protein. So here's our protein on the surface, here's the epitope, and what that does is it displays the epitope to T cells so that, like we see here, the T cell can bind and mount an immune response. Cytokines are basically just chemical signals from one leukocyte to another. So it's a way that different leukocytes communicate with each other, and interleukins are one type of cytokine. Okay, so cytokines are chemicals that are used to communicate between leukocytes, and interleukin is one type of cytokine in particular, and you're going to hear about interleukins a lot because this is how lymphocytes and antigen-presenting cells communicate with each other. So here's an antigen-presenting cell in the form of a macrophage. Here is a lymphocyte or a T cell, and they are able to communicate with each other through these cytokines called interleukins. All right, so practice questions for this section. How does adaptive immunity differ from innate immunity? Remember, we talked about those three distinguishable features. We talked about, let's go back here to the beginning. Specificity, memory, and systemic effects versus local effects. How does humoral immunity differ from cellular immunity? And again, like I said, we're going to go into much more detail in the next couple of slides, the next couple of chapters, but it is important for you guys to have some idea here. And so let's go back to that slide. That's going to be this one right here. Remember, cellular immunity employs T lymphocytes to directly attack foreign cells in places they are inaccessible to antibodies. While humoral immunity uses B lymphocytes, it doesn't attack cells but uses antibodies to target pathogens or microorganisms that live in the or are within the extracellular stages of infection. Okay, contrast active and passive immunity. So we talked about vaccines versus colostrum. Provide artificial and natural examples of each. So make sure that you're able to distinguish between those examples. What types of structural components and properties distinguish antigenic molecules from those that are not? So remember, we talked about the importance of, let's go up here. Of these antigens or these components of the plasma membranes being much longer than something more simple like a glucose. So they're going to be proteins, polysaccharides, glycoproteins, glycolipids, and that's important because that length of the molecule or size of the molecule is what allows it to be unique. Okay? Describe the structure of an antibody monomer. So we talked about the 4 chains, what part of it binds to the antigen. So remember that yellow part, that one from each of the 2 types of chains comes together to form a binding site for the epitopes or the active part of the antigen. What is an immunocompetent lymphocyte? So what does it mean to be immunocompetent, and what are the processes that a lymphocyte has to go through to get to that point where it is considered immunocompetent? What role does the thymus play in the life history of a T cell? So this is sort of a redundant question in that you may have answered it when you answered this question, but what happens in the thymus that causes a T lymphocyte to become immunocompetent? What are the two tests that a T lymphocyte has to pass? What role does an antigen-presenting cell play in the activation of a T cell? Okay, so we talked about here what's actually happening in this antigen-presenting cell that allows the T cell to bind to it and stimulate an immune response. All right, so I'm going to stop this video here, and then I will post a second one on the next couple of chapters.